Well, thank you all. Thanks for being here. I'm excited to see so many friendly faces. I appreciate you coming. For those of you guys that know me well, you know that I love to talk about this sort of stuff, so I'm going to try to dial it back a little bit, not go into too many tangents, things like that. But give you an idea of an overview of what I'll be talking about today. I'm just going to try to orient everybody because I know that everybody doesn't spend most of their time thinking and talking about this stuff. So I'm going to give you an idea of the underlying psychobiology and why we're studying hormones, the value of studying hormones in general. Give you a brief primer on the stress response and associated systems, things that I'm using to look at some social behavior, and give you just also a brief overview of how life history theory is used as a framework for understanding the interactions between our biology and our sociality. And then finally talk to you about the research that I've been doing since I've been here on the psychobiology of relationships. I divided up into a couple different ways. The first is looking at individual differences in sensitivity to context, how we differ in that. And then looking at the psychobiology of specific relationships, some that are understudied. So the first is female relationships. And then looking at within families, looking specifically at the role of fathers. So, why do I care? Why do you care? Why should you care about hormones and things like that in general? Well, first of all, our human sociality is a bit of a mystery. It's a bit of a paradox in that our relationships are our most salient stressor, but they are also our most powerful buffer or antidote against that stress. Some research that has used the 10-item Adverse Childhood Events Questionnaire has really borne out this idea of early social relationships or context within families and things like that having long-term impacts. These items can be shown to be predictive of physical and mental illnesses, also health risk behaviors, and this is over and above socioeconomic status. So children, for example, in preschoolers with more than 3 ACEs show poor academic outcomes. They show poor attention and have social problems and aggression. These events are so powerfully predictive of long-term health outcomes that now healthcare professionals are starting to use these or assess these in their routine medical assessments because they can help them know what type of assessments their patients may need and how to treat them. So it turns out that the Beatles were right. All you need is love. They were a bit too idealistic, though, as that is difficult to attain, right? If that was the case, we would all be healthy and happy. This quote from E.O. Wilson has always stuck with me from undergraduate all the way through my graduate training, in that what are humans ideally suited for? And specifically related to some of the topics I just covered with ACE scores, why do we have a stress response system that is so open to developmental context that it could then be damaging? It doesn't seem to be very adaptive. So hopefully I will help to answer some of those questions today. But first, let me give you an overview of the systems that I'm studying and some of the associated biomarkers, just to orient everyone. So first, starting with the autonomic nervous system, and most of you guys know that it's further broken down into sympathetic and parasympathetic nervous systems. This is our fight or flight response. This is our first responder, the thing that comes first online when we experience a challenge or a stressor. The biomarkers that I use to assess these are salivary alpha amylase and sometimes respiratory sinus arrhythmia. Then moving into the hypothalamic pituitary adrenal axis, this is also related to fight or flight, but this is a slower responding system. The system is also particularly responsive in humans to psychosocial stressors. It also regulates attention, memory, and then also neural reorganization. I should also say that this system in particular has a diurnal pattern in that it is highest, your cortisol levels are its most active, highest in the morning, and then it tends to decline throughout the day. That'll be important in just a bit. And then we also need to look at hypothalamic, pituitary, gonadal axis. These are all systems that innervate within each other, but these are regulating the gonads, so testes or ovaries, regulate, not surprisingly, reproductive behavior, parenting, and also competition. Associated biomarkers are testosterone, estradiol, and progesterone. And we can't leave out oxytocin. It's produced by the pituitary gland. Oxytocin has a role in regulating reproduction and parenting. It also has a role in affiliation or bonding. And it also helps to interact, it interacts with the HPA axis to reduce stress responses. And then finally, I also examined the immune system and immune response to inflammation in particular. And these systems are all interacting. In general, what you get when you get an acute stress response is you have initial activation of the immune system. Then once stress becomes a bit more chronic, sometimes depression of the immune system or other types of interactions. So this is, by the way, not an exhaustive list. This is just a general list of what we're doing here. The stress response systems in particular really reflect an organism's priorities. You can't get more basic than fight, flight, eating behavior, and reproductive behavior. Those are the core sort of priorities for an organism, and these systems help to regulate that. These are the systems that have kept us alive for eons. Switching gears a little bit to talk about life history and life history strategies. Life history theory says that organisms have limited time and energy. I think we would all agree with that. And that we need to have trade-offs then. We need to allocate our time and energy between growth and reproduction, mating, and parenting. So life history strategies among different species are broken up a couple of different ways. They're on a continuum from slow to fast. So species that have fast life history strategies tend to have a high growth rate. They mature fast. They have a lot of offspring, and they provide little or no direct parental care. So these are species like goldfish and things like that. This is for my son. He wanted me to put a picture of a goldfish up here. That's his favorite slow life history strategist. So on the other end of the continuum, we have, fast life history strategist. We have slow life history on the other end of the continuum. This is characterized by a slow growth rate, large body sizes, long lifespans, also very intensive parental care. So these are characteristics of species like elephants, but also humans. The risk in a slow life history strategy is dying before reproduction. Okay, you're intensely, investing in very few offspring that could die before they get to maturity. So these are the trade-offs that we're interested in, and we have interspecies trade-offs as well. But my question is, essentially, how did she get here? This is a species that has single offspring at a time. These offspring are very expensive to produce. They have big brains. They take decades to mature, and those brains require lots and lots and lots of calories to grow. She can't do this on her own. In an ancestral environment, there was no way that moms could provide alone for their offspring. This also doesn't seem to be a design of a species that would eventually take over the planet, right? So how did she get here? How did she do this? The answers are in a number of our unique relationships. So we have family relationships, we have bilateral extended kin networks that facilitate things. We also have form alliances with same-sex peers, so these are friendships. And then finally, we rely heavily on pair bonding in our mating relationships. So, in looking at the psychobiology of a relationship, so this underlying trade-offs between sociality and our biology, we see that for humans anyway, the common theme is intense sociality. We have a number, like I said, of unique relationships, cooperation as well, even among non-relatives. This is actually relatively rare in the animal kingdom. So hormones can help us understand life history trade-offs. They help us also understand individual differences among us and individual differences in sensitivity to social evaluative threat. Social evaluative threat and exclusion are where I focus a lot of my research, and these reliably activate all of the systems. The HPA system, the HPG system, and also immune system is also activated by social evaluative threat. So these are the hallmarks of systems that were shaped by natural selection to help us navigate cooperation within our relationships, but also competition. So first, I'm going to talk a little bit about developmental context and how that's going to be related to how we adjust our stress response systems to better adapt or better calibrate it to our environments. So this is borrowing from the biological sensitivity to context model, and in it, what it shows essentially is our developmental context matters, and it matters a lot to our stress response systems. They're very plastic and open to modification, particularly during certain times of development, infancy in particular, and also later on in adolescence. And it results in different patterns of reactivity. So in one end, you can have a very sort of low stress safe environment that results in a phenotype that is sensitive and responsive and open to that context. It's not necessarily a bad thing. These individuals are able to take advantage of social support networks and allow that to further create opportunities and things like that. You have a buffered response when you have moderate stress. So these are individuals that have had moderate amounts of stress throughout their lifetimes. They're not very reactive, but that actually has a trade-off in that they're not always, sort of activating their stress response systems and wasting energy, so to speak. Those coming from a more unpredictable environment, so say parents that are unpredictable in their investment, parents that were there sometimes and not, that tends to result in a phenotype that is more vigilant, okay? So you have high stress response systems. The cost is that you are consistently activating the stress response systems. The benefit is that these individuals are then keen to be able to take advantage of challenging situations and meet those challenges and with some extent of efficacy. So I'm going to focus on this model. We also differ, though, in our individual responses to context and how modifiable our systems are. So the first series of studies I'm going to tell you about are looking at these individual differences in response to social rejection. In particular, we were interested in individual differences genetically, but also in trait rejection sensitivity. So our focus here was on one particular oxytocin receptor polymorphism that has been associated in the past with oxytocin production primarily, but also cognition and social behavior. It has a role in regulating stress responses. Those individuals that have the GG genotype as opposed to A allele carriers, that's associated with more pro-social behaviors, things like empathy, sensitive parenting. And we posited that this increased sensitivity to the social environment would also be related to rejection sensitivity. Okay, so if you're open and responsive to the environment, those can be very good things. You can be very pro-social and empathetic, but it also can put you at risk for being sensitive to rejection. So the first question we asked was, is rejection sensitivity related to baseline or post-evaluation cortisol levels? And then does that interact then with genotype to influence cortisol recovery? Remember, cortisol takes a while to sort of peak and then respond afterwards. So we had 94 undergraduates, and the tasks that we gave them were that they were asked prior to coming into the lab to think about a salient social problem. So something that was important to them, things like a lot of these came up were things like addiction or homelessness, things like that. And they were to propose a solution to the problem. The story that we gave them was that they were competing for a scholarship, a $150 scholarship, and that they would then come in and write in the lab. So after completing questionnaires on rejection sensitivity, they were then told that they would be evaluated by a committee of their peers, and these peers would be the ones that would select them for this particular scholarship. In fact, they were randomly assigned to either scripted praise or criticism. Don't worry, we debriefed them, they're fine. All right, so what we found was that they were actually, first of all, to answer the first question, rejection, rejection sensitivity itself did not predict cortisol responses in any way. When we include genotype, we get some interesting interaction. So at post-evaluation, right after this evaluation, we really see no differences among the different groups. But post-evaluation, what we see are those actually that are low in rejection sensitivity, but have the GG genotype being the highest reactors, along with those that were high in rejection sensitivity, but A allele carriers. Those with more risk factors actually had this sort of declining result after evaluation. So these results suggest that there's a curvilinear relationship between social salience risk factors and response to social rejection, such that those that are at moderate risk are actually the most likely to mount a cortisol response. We can look at this another way by looking at individual differences and response to public speaking. Apparently some people find this stressful to speak in public, particularly when they're being evaluated on it. So I don't know what they're thinking, but that's what they tell me. So in this study, we were looking at undergraduate students in their speech communication classes. So prior to participating in their speech that was a major component of their grade, they completed questionnaires on communication anxiety, rejection sensitivity, and social interaction anxiety. We expected that that would augment their stress response. So we looked at both stress response systems, the ANS system and also the HPA system. And we were interested also if this is affecting their immune response. So do repeated levels of public speaking, social evaluative threat, does that influence our immune system? Those of you guys that have gone through finals or graduate school in general know that when you get really stressed, what happens? You get sick, right? Usually right post-stressor. That's because these systems are really interacting with each other, and you can have a suppression of the immune system with chronic stress. So for this study, we had 95 students that were participating, the majority of them female. And the social salience risk factors that we identified, again, were being high on communication anxiety, rejection sensitivity, and social interaction anxiety. We also included males in the model. Males tend to be, as I'll tell you in a minute, more reactive to this task. So before I go on, I'll tell you that there were no sex differences. So we didn't find any sex differences there. That was dropped from the model. These are the responses for the overall group, just showing that giving a speech is actually pretty stressful for students. So you see this cortisol reaction where this is pre-speech, right after the speech, and then 20 minutes following. You see this nice cortisol peak right then. Alpha amylase, again, being the faster to respond, is peaking right after the speech. And then with IL-1 beta, it's a measure of inflammatory response. You see sort of an anticipatory response right before the speech, and then it's declining there afterwards. When we break this down into risk factors, first of all, rejection sensitivity didn't predict anything, so that was, again, dropped from the model. When we're looking at inflammatory responses, we see this nice linear pattern with those with no risk, not really mounting much of a response, moderate risk, meaning they're high in either communication anxiety or social interaction anxiety, had a modest response, and those at highest risk with both elevated had that higher response. This was also somewhat true for cortisol levels, and this is all cortisol increase, so the total increase from baseline, and this is total cortisol output. And we see a consistent pattern here where with low or to moderate risk, we really don't see a big response. Those that had both communication anxiety and social anxiety elevated had much higher risk. So this suggests that there are some people that may be more responsive or more sensitive to social rejection than others, and we can predict this by looking at some of their traits. So another way to look at individual differences is to look at sex differences, and I'm borrowing from Laura Stroud's model here, where she divides these up into social rejection stress and achievement stress. So we're looking at basically social exclusion is a primary tactic and a common tactic for females. It represents sort of an implicit status threat, And it functions to disrupt the social ties and status of competitors. Okay, so we would expect then, following from that, that women would be more responsive to social exclusion as opposed to achievement stressors. Achievement stressors represent an explicit status stress threat, and males tend to be more reactive to this. These effects are pretty robust across a number of different labs. I should say also that there's no difference between reported stress in both of these. So both men and women say that both of these stressors are really, really stressful. They really hate it. But what we do see are differences in their physiological stress response. So this can be extended to eating behavior, and this is a project with my graduate student Nikki Klaus's thesis, and we were interested to see if this could be sort of shown in the lab. We know that stress and eating behavior are linked. For example, after today, I'm much more likely to go for the chocolate cake and probably will pass over the broccoli. Even though I really like broccoli, cake sounds a lot more delicious to me right now. We also know that chronic stress is related to the consumption of highly palatable foods. Okay, so these are things like the chocolate cake. They're delicious. They have that sort of inherent fat and sugar drive. And most of the work in this area has focused primarily on women, and so to such an extent that a lot of the literature actually suggests that this is a female typical sort of response or problem. And we reason that it may be actually due to the stressors that were evaluated in a lot of this research. These are primarily relational stressors that they're looking at. So we employed Stroud's model, and we had 168 undergraduates participating. They were randomly assigned to different groups. So in the achievement stress group, what they did is and all of these groups had Confederates that were assisting with us. And in the achievement stress group, they witnessed a Confederate perform very well, and then they were asked to do the same tasks. The tasks were memorizing a passage of Paradise Lost. If anybody's ever read that, it's not super easy to memorize. Also, the other task within that was performing a New Roman math system that was essentially an impossible task. They couldn't solve it, but they saw the Confederate that went right before them and did very, very well on these. Of course, our Confederates had all of the answers. So in the social rejection stress condition, they came in again with Confederates, and they were told that we were interested in how people got to know each other and how they exchanged information. So they were asked to undergo a getting to know you task, and in this task, the two Confederates appeared to connect a lot better with each other, and they systematically excluded the participant by doing things like, really sort of, basically saying, oh, well, I like to do this, you don't like to do this, and sort of, looking at each other basically like, oh, we're very connected, we have so much in common, we should get together after this, those sorts of things, and just ignoring the participants, or maybe even devaluing their interjections. In the control condition, this is one thing that Stroud's work has not done, is have a control condition in this, and we thought that it was important. And we wanted to control for just interacting with a stranger in general. That could be stressful, right? And so they were interacting with our Confederates in the lab, and the task was to design a recreation center. I've used this for a controlled task for a number of studies. You'll hear about it again. And in this particular task, they just designed the the amusement park together, and they were given a list of features that it had to have, and they just sort of designed it on a piece of graphing paper. So this is the basic sort of protocol. They came into the lab. We got physiological measures on them, and they also, by the way, the important thing with this task is that this happened during lunchtime. So we had them come in without eating. We said you can't eat 2 hours or so before coming in, and because we wanted them to be hungry, we told them it would interact or influence the physiological measures we were taking, which it actually does, so we weren't lying going there. And we then had them undergo the task. And then afterwards, we said, well, there's this other part of the experiment, but there's not enough room in here. You need to go wait in this other room. And in the other room, the food room, we said, well, we feel really badly because you haven't eaten, and so there's this food that's left over. Just have, whatever you want. We'll be back in a few minutes. And so that was our other dependent variable there. So we looked at salivary cortisol and RSA. And then we measured their post-stressor food consumption. The food was divided into a couple different groups. So we had highly palatable sweet foods, things like cookies, highly palatable salty foods, potato chips, and nutritious sweet foods, so things like strawberries and grapes, and nutritious non-sweet foods, again, broccoli. I don't know why I keep coming back to broccoli, but there's broccoli there, celery, things like that. And then bland foods, things like white bread, crackers. And so what we found were probably the most beautiful interaction effects I have ever seen in my life. So this is highly palatable food consumption. This is the number of calories consumed. So what we see here is the males consuming much more highly palatable food in the achievement stress condition compared to the females. And then that pattern is reversed, where females are consuming more in the social rejection condition. We see no differences in the control condition. And in the control condition, we see that they weren't necessarily as stressed, and they weren't eating as much. We also assessed their hunger, by the way, and so there were no differences across the group in that. This is with respiratory sinus arrhythmia. Remember, negative numbers on this means more responsive. So we see males, again, being more responsive to the achievement stressor, and females more responsive to the social rejection stressor. And the exact same pattern for cortisol as well. So we have a higher cortisol output for males in the achievement stress condition, and more for females in the social rejection condition, and no differences in the control condition. So the take-home point here is that men also engage in stress-induced eating. It depends on which kind of stressors that you're looking at, and it may be, like we thought, is that these other factors, these other studies that have looked at this didn't really take into account the different types of stressors that might initiate stress-induced eating. Okay, shifting gears a little bit here to focus on specific types of relationships. And so one of the questions is, why do we form relationships at all with same-sex peers? It's a difference between us and other species, and many other species anyway. And the other question is, how do these relationships form? So what are the mechanisms that are behind them? For this, I am borrowing from Shelley Taylor's seminal tend and befriend model, which essentially she said that women's relationships, instead of being characterized, or women's stress responses, instead of being characterized more by a fight or flight response, are more characterized by tend and befriend. And what she meant by that is that when we experience psychosocial stress, and for women in particular, gaps in social relationships will tend to initiate that. This pulls in oxytocin and will initiate affiliative efforts. Okay, so we're trying to get social support. We seek social support and trying to get that. If these this affiliative efforts results in negative social contacts, then we have an exacerbation of stress. If it results in positive social contacts, so good social support, we see a reduction in stress overall. So within this, we know that girls and women often form, these affiliative efforts. What they look like are primarily dyadic self-disclosure. So what that means is looking at different, sort of exchanging personal information back and forth. And some of the women in the audience, you know what I mean by this. Sort of, talking about, talking about your problems, talking about your past, events and relationships in particular, things like this. We get uncomfortable when the balance is a little bit off. So when you've shared a lot of information and people aren't sharing back with you, it makes you pretty uncomfortable. Get to that later. But one specific type of dyadic self-disclosure, or one extreme form, is co-rumination. So this is Amanda Rose's work that I have been using for quite some time. It's an extreme form of self-disclosure, and it has 4 basic characteristics. The first is excessive rehashing, and these are sort of within discussions where they're talking about problems that they're having, trying to seek social support. They're rehashing the problem over and over, speculating about the causes and consequences of the problem. There's a pretty high focus on negative affect. So how bad or upset the person with the problem feels, how bad or sad the friend feels for that person. And then there's a component of mutual encouragement. So there's both of these friends are really trying to keep each other talking about the problem, keep rehashing, and those sorts of things. So within this, it occurred to me that there are some trade-offs, and some of these are seen in the literature, and that co-rumination actually results in some benefits. So we see increased feelings of social support. So that works. Increased relationship quality. These relationships are pretty high quality if you can tolerate the negative affect focus. And this may serve as a potential ally in times of duress. So this is someone who knows a lot about you, is invested in your problems, and things like that. And some of the questions that we have been asking are, does it serve a bonding function? Do people that co-ruminate, are they closer? Are they more bonded together, and how can we assess that? It also has some costs, though, in that we see increased internalizing symptoms. Amanda Rose has called this the cost of caring, in both friends. So they have more anxiety problems, more depression symptoms. We see some stress system activation, and potentially vulnerability to relational aggression. So you're really, sort of sharing a lot and exposing yourself. That can then be turned if the relationship is terminated. That information can be shared, and that won't necessarily be in your favor. So it occurred to me that co-rumination really occupies this space in between negative social context and positive social context. So it has, it's supportive, but it has a high negative affect focus, which negative affect focus in general will initiate a stress response. And so the question is, does it, sort of increase negative affect focus will then return and sort of perpetuate this system of increasing allostatic load? Or eventually, does this turn into a situation where you can have attention to solutions and bonding through the stress system activation, and then eventually reduction of stress and adaptive reorganization of neural networks? So that is what we have been testing. And in the first of the series of studies, we had dyads of close female friends, best friends, BFFs, come into the lab, and they were randomly assigned to either a problem discussion task in which they listed typical problems that they talk about in their relationship, not problems within the relationship, problems outside of the relationship that they're bringing in to provide social support, or a control task. So this is, again, the same control task that I use in a lot of studies where they're, in this study they were designing a recreation center together. So it's very, still social, but very neutral in content. So we measured cortisol levels before and after their interaction, and we videotaped these. I was concerned at first that we couldn't get them to really co-ruminate on video. I was real wrong about that. They will share all sorts of information, so that really reduced my methodological questions there. So we had 48 individuals that were participating with their friends, and what we found is that we had a condition by observed co-rumination interaction. So those in the problem discussion group did co-ruminate more, and we found that negative affect focus in particular was the best predictor of cortisol reactivity. So the sort of focus on how upset the other person felt and things like that was really initiating the stress response. We followed this study up in looking at dual stress system activity. So my question was, well, we know about the HPA axis with this. Do we have a sympathetic nervous system response? And what we found is this is the problem talk condition. This is the control. And so you even in the control condition, as we found later, they still co-ruminate a little bit at least. And it was more, sort of complaining about having to be there and have this research credit, and wasn't this a boring task, and those sorts of things. So that's okay. We get a little bit of stress response activation when negative affect focus even in that group gets high. But what we found for the sympathetic nervous system in particular is that it takes higher amounts of negative affect focus to really initiate that system. The other thing is that this dual system activity may actually provide the mechanism between the relationship of co-rumination and internalizing symptoms. So this pattern of responding is consistent with increasing internalizing symptoms, and so this may be, again, the mechanism by which that happens. So we then asked as to whether, this question of bonding, okay? So are these individuals eventually, sort of going to, get to a situation where they're finding solutions and things like that, but also are they closer together physiologically? We know that within the psychobiology of relationships, we see, one of the things that my research has really been focusing on is this issue of adrenocortical attunement. Okay, so synchrony or coordination within individual stress response systems with another individual with which they are interacting. We know that this is well documented in family relationships and also romantic relationships. Within mother-child relationships, we know that mothers that are more synchronized with their infants are more sensitive and responsive to them. We also know that those infants have better emotion regulation. In romantic relationships, most of this work has been done in conflict discussion tasks, and we see that couples who are more coordinated, even if that means both of them are being very reactive to the conflict, that actually predicts positive relationship outcomes. Okay, so when they are discordant, when they're asynchronous, that is actually a risk factor for divorce. Our question was, how does this happen in friendships, or does it happen at all? Is it going to, sort of transcend to this other type of relationship? So again, with this study, very similar methodology. We had 74 individuals. They're all coming in with their best or close friends. We had a pre-task assessment, and then we had a warm-up task. And their experimental task, again, was the problem discussion or the control task. We took saliva samples immediately after the task and then separated the dyad to separate rooms so that they couldn't interact, and then had a follow-up 20 minutes post discussion. And our questions were basically, if this is related to interaction, these friends should show attunement or synchrony in their cortisol responses when they're interacting, but not necessarily after they have been separated. And that's what we found. So pre-task and post-task, they were correlated with each other, and then follow-up as a group, they were not. And then the second question was, does pre-task adrenocortical attunement, so the levels, the extent that they were similar pre-task, did that then predict their level of co-rumination? Does that sort of set the stage for them to be able to co-ruminate more? And it turns out, yes, it does, both for overall co-rumination, but also for each of the components of co-rumination. Then we asked, well, does the level of co-rumination during the discussion, does that predict higher levels of post-task attunement? Does it sort of serve this function of sort of keeping them together, keeping them synchronized with each other? Yes, it does. And then does co-rumination then, those with higher levels of co-rumination, are they more likely or likely at all to be correlated at attunement? And we found that that was the case. So those that had higher levels of co-rumination within their discussion task, and this was irrespective of whether or not it was in the control task or the problem talk task, that predicted higher levels of attunement at follow-up. So it appears that, I can't say the word, excessive complaining with negative affect focus is bonding, at least for women. I almost said it. Okay, so switching gears a little bit and looking back at this sensitivity to developmental context, we see that you have different patterns of sort of social interactions, particularly with important relationships. These are primarily parent relationships and extended kin networks. And I'm interested in sort of focusing on this level. So this sort of unpredictable early environment, particularly with parents, and this sort of vigilant responding. Is that then going to translate into, from the family relationship, we know that that translates into individuals, but does that then translate into other types of relationships? So the way that you respond in other relationships, such as friendships. So specifically, we ask, and I mentioned before, fathers are in general less studied than mothers. I'm coming to find out why that is now, because dads don't come into the lab and don't sign up for studies. But so in this one, we ask young women, college students, about their relationship with their father. And we know that paternal investment is common for humans, but it's also more variable than maternal investment. And so in some ways, it may be a better predictor of differential outcomes than is the relationship with moms. We also know that it is a salient life history cue. We know that it predicts romantic relationships and things like that later on, particularly in cultures that are Western, educated, industrialized, rich, and democratic. It doesn't actually, some of these findings are not found in other cultures where you have a more extended kin network. But paternal investment or father presence or absence is associated with age of menarche, age of first birth, and mating strategy. So it is calibrating life history strategy either towards slow or fast. And it is associated, like I said, with stress responses in romantic relationships. And so we're asking about friendships. Is this going to translate then to friendships? So in the first study, we examined the diurnal pattern. So this patterning of waking up and then sort of how you're supposed to have high cortisol levels if your system is functioning well at that point, and looking at it over the course of the day. And we found that father-daughter relationships that were characterized by more rejection, chaos, and coercion were actually associated with lower morning cortisol levels. This is not a good thing. Okay, you need this system to be active and responsive, and this is actually indicative of some disease states in psychopathology. In study 2, we ask girls to come in with their friends, and we assess their relationship with their father, or at least their reported retrospective relationship. And we found that those with warm, consistent, supportive fathers had lower baseline, lower pre-test cortisol levels, and a more attenuated or reduced cortisol response to co-rumination. More negative and harsh and consistent fathers were associated with enhanced stress responses to co-rumination. So this is showing, in a sense, that this is translating into a number of different relationships. They're using this developmental context then to try to predict future relationships and how to respond in those. We also see that father-daughter relationship quality was not associated with salivary alpha amylase levels. So it is not necessarily reactive with the autonomic nervous system. All right. More on bonding, more on family relationships in general. I'm interested, this previous study was looking at, sort of a retrospective account. So these, young women were asked to think about their relationship with their father during childhood and sort of report on that quality. And that's, that's fine, that's the best of at the moment, but I'm really interested in looking at these intergenerational links between stress system responding and stress system activity as they are occurring. And so again, looking at attunement or coordination of stress system activity, looking at a couple of different time points, critical or sensitive periods in development. So the first is infancy. We know that infants are remarkably attuned to their social environment, and they use caregivers' behavior as a clue to their social climate. So is this a safe environment? Can I trust individuals? Can I generally depend on others? Or is it more unpredictable? Do I need to be more vigilant in my responding? We also know that adolescence is another sensitive time period. We see an increase in brain and stress response plasticity during this time period. We also see an increased sensitivity to psychosocial context, particularly peer context, but also parental context as well. Previous research has shown that adolescents are attuned with their mothers over the course of the day. So they show consistent and similar diurnal patterns given that they have enough interaction. So adolescents, as much as they're trying to push away, are still sort of physiologically linked, at least with their mothers. And so one of the questions that I ask, and this is a study that was in culmination with my colleague Mike Chris and Amanda Morris, and we were interested in looking at adolescent girls during times of conflict. Adolescence in general can be a time of conflict, particularly for mothers and daughters. And so we were asking if if they were attuned to their mothers during conflict, and if that attunement was related to genotype, if it was related to the amount of oxytocin that they generally produced. We had 171 dyads participate in this study. These were collected in Tulsa, and these girls were ranged in age from 12 to 16 years old. I want to point out a couple of unique features of this sample. It was primarily African American, and it was also primarily low income. The reason that that's important is that these families were families that were likely experiencing a number of chronic stressors in their life. So these are families that are really trying to deal with a lot of challenges anyway, irrespective of the challenge of raising an adolescent daughter. So what we did with this is we had them come into the lab. They did a number of other tasks. This is the section of a larger project. But with this, we had them engage in a conflict discussion. So they talked about a conflict that they were having, and we had them discuss it in the lab. And our question again was, do we see the synchrony or attunement related to that during this discussion? Just looking at simple correlations, we found that that was the case. So moms and daughters' cortisol levels were correlated, at least at both time 1 and time 2. And then accounting for a number of other factors that influence cortisol levels, things like time of day, incomes to needs ratio, and things like that, we found that mothers' cortisol, post-test cortisol levels were predictive of adolescents' post-test cortisol levels. So this is showing that they were attuned to that particular context. So they were tracking each other both behaviorally and physiologically as well. The next question was there attunement in response to the task or change in overall cortisol output? And does genotype matter? Yes and no. Okay, so parent cortisol change was predictive of adolescent cortisol change, but we did not find an effect of genotype in the study. So we think that probably the more important thing here is the interaction and those sorts of sort of tracking each other's behavior and things like that. And at least for this particular SNP, it wasn't predictive. All right, so turning to infancy, so that's in adolescence, and turning to infancy for a minute, we're asking what factors influence cortisol attunement in infants. First of all, mothers. Mothers are really, really important. Infants know to attune to their mothers. They're their best sort of indicator of the social climate. But mothers are also influenced by a number of other things, and one of those primary factors is social support. So moms that have more social support are more sensitive and responsive to their infants. Mothers that have less are not. Infants are also influenced by their quality of alloparental care, so the quality of care that they're getting from fathers, potentially from grandparents, siblings, and maybe unrelated caregivers. And socioeconomic status matters as well. So infants have no idea about money. They don't have any idea about resources, but they do seem to track their parents' response to some of these economic stressors as well. So in this series of studies, we were looking, this is a preliminary investigation of looking at the effect of mother's social support and social support networks on her and her infant's cortisol synchrony, and how that was related to the amount of infant-directed speech. So we were interested in how much mothers were talking to their infants, and that is predictive of infant cognitive and language outcomes. So we had mothers and infants that ranged between 4 and 10 months. Moms were approached at public places, and so basically we saw a mom with a baby and we made a beeline over there and said, hey, you want to fill out questionnaires and give us spit? And some of them did. Okay, some of them thought we were crazy, and that was okay. So they filled out questionnaires, and this took on average about 20 minutes. If anybody has ever spent any time with an infant, they know that they really don't like their moms not attending to them, especially for 20 minutes. That's a mild challenge for both mom and infant. So we use that to serve as a challenge. The sample demographics were, relatively, sort of white middle class. Most of them were married. Most of them were college educated, and the average amount of children they had was around 2. We calculated synchrony or attunement by looking at, or by subtracting, taking the absolute value of the mother's cortisol levels minus the infant's. So it's important when you're looking at these results to know that those that are closer to 0, that indicates more synchrony. So closer to 0 is good in this case. And looking at the pattern of correlations, what we found is that, first of all, infant cortisol levels were related to couple satisfaction. So the more satisfied mom was in her relationship with her romantic partner, the lower the infant's cortisol levels were. Okay, so infants are not as reactive in that context. We also found that was related to mother-infant cortisol synchrony, and that mother-infant cortisol synchrony was associated with infant-directed speech time. So moms and infants that were more coordinated in their cortisol, moms basically spent more time talking to their infants and things like that. So this can be sort of a mechanism by which moms that are sensitive and responsive can be, and then lead to cognitive and social outcomes for their infants. So we're asking, we know that dads matter, we know that mom's social support system matters, but one of the questions that we have been asking recently is how much do fathers matter? How much is it dads versus how much of it is just social support in general? So in this project, this is a project that is funded by both OCAS and CIRCA, and we are looking at the pattern of hormonal coordination in fathers as it's related to mother-infant adrenocortical attunement and important infant outcomes. We're looking at language outcomes, but also cognitive and health outcomes. We're following families from pregnancy through the first 18 months of their infant's life. We're attempting to recruit a high-risk sample simply because there is less known about, again, how these families are negotiating these challenges outside of the family and within the family. So we're interested in sort of determining that. We're taking monthly assessments of, sort of general social climate. How are things going? What is your level of social support? Alloparental social support in particular. We're developing a new measure. Hopefully it'll become a new measure of looking at that. So this is looking at not only how much dads are contributing, but also other family members and perhaps even, friends and others that are maybe unrelated to the infant. We're assessing developmental milestones. So how on track these infants are, and then health of the overall family. We have home visits when infants are 4, 12, and 18 months old. And we're assessing the whole family for cortisol. We're looking at, again, at synchrony or attunement. We're looking at CRP, an index of inflammation. And then in fathers, we're looking at their testosterone response to these interactions. And in mothers, we're looking at progesterone. And the basic theoretical model here is that we know that maternal sensitivity is related to mother-infant adrenocortical attunement. Our question is, does father involvement moderate that association? Okay, does father involvement, sort of switch the pattern of responding there? And also, is father involvement directly associated, this other arrow here, directly associated with mother-infant adrenocortical attunement? And then how does this entire family hormonal profile, so testosterone, cortisol, progesterone, and CRP, how is that influencing the relationship between mother-infant adrenocortical attunement and language and cognitive outcomes? We have some exploratory aims as well, where we're looking at this triadic adrenocortical attunement to see to the extent that the 3 members of the family are interacted. I forgot to mention that at the home visits, we're having them engage in a series of tasks where we're video recording so that we can link their behavior then to their physiology. So very, 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 I can't express this enough, very preliminary results. We don't have a large sample size that we have everything analyzed on right now. But we know, this was interesting to me, that father's couple satisfaction, so the extent that he is satisfied in his relationship with his partner, is positively related to gestational age. So infants being born later in pregnancy. This is a good thing for infants. They need a little bit more time and things like that. It's also associated with his lower baseline cortisol levels and mid-visit cortisol. So this is the effect of, positive relationships are moderating his stress response. For moms, her satisfaction with her partner was positively related to birth weight, so they had heavier babies, and negatively related to total perceived stress. So this is showing us that dads really are, this, to the extent that this relationship is a high quality relationship and supportive, then at least reportedly mothers are not as stressed. In case you were wondering, mom and dad's couple satisfaction was correlated pretty highly across a lot of these measures. Okay, so future directions. Looking at one of the projects I'm really excited about, and this is in conjunction with my colleague Mike Chris, is looking at discrimination stress in emerging adults. And we're looking at the pattern of responding both to typical lab stressors, things like the Trier social stress test, but also looking at their diurnal patterns. So their patterns just, sort of daily functioning of the stress response system on a typical day. We're just looking at the data now, so I can't present that, but we're excited about it, and this is going to serve as preliminary data for 15 proposal in February. Second line of research is examining men's developmental context. There's not a lot that's been done on looking at how men's developmental context is related to both their mating and parenting strategy, but also some of these associated hormonal correlates, particularly in response to infant cues. So in this study, this is my graduate student Ray Garza's project, where we're looking at eye tracking, and we're assessing how fathers are different when they're looking at their own infant versus another infant. And so I'm excited to get that project up and going. And then probably the most exciting is further examining female friendships and also rivals, so female-female competition and the hormonal profile related to that. So we're looking at both, sort of responses to competition or competitors, and then looking at the role of friends in buffering some of those costs, or having allies. This issue of, do we like her? Okay, is this going to then buffer some of those costs related to competition and make one more effective? So in conclusion, how did she get here? It turned out from a little help from her friends, her mates, and her family. I want to acknowledge some of the people that helped me get here. I have been really, really lucky to have a number of really excellent collaborators that have been supportive and also have pushed me a little bit to learn new things, and pushed me a little bit beyond my comfort zone, and the results are pretty fantastic. I also want to acknowledge both my current and former graduate students, maybe future too, and they have been really integrative and sort of helping again push these projects forward, but bringing their own ideas in and allowing us to test some really novel and cool predictions. And I also want to thank this collection of graduate students that I have borrowed from other labs. Thank you, mentors, for letting me borrow your graduate students. A lot of this requires a lot of manpower behind it, but I'm fortunate to have these collaborations and have these collaborations go well. I would love to acknowledge, I started to list all of the undergraduates that have worked in my lab, and they could not fit on the slide. So I'm just going to say thank you for my undergraduate research assistants. Again, a lot of this work has required a lot of manpower, and I'm really fortunate to have some really nice junior scholars that have assisted with that. So, questions?